Hi my dear students, welcome to my channel NEET Biology Diagram Based Technical Terms Part 4. Today the topic Respiration. Important terms are given. Number 1, Aerobic Respiration. Aerobic respiration means oxidation of complex organic molecule in the presence of oxygen with the carbon dioxide, water and energy. Cellular respiration may be classified into two types according to its dependence on oxygen. Number 1 aerobic, number 2 anaerobic. In aerobic respiration, organic food is completely oxidized with the help of oxygen, that is terminal oxidant, into carbon dioxide and water. Aerobic respiration occurs in mitochondria. 686 kilocalories or 2870 kilojoule of energy per molecules of glucose is liberated during aerobic respiration. The earlier estimate was 673 kilocalories. C6H12O6 plus 6O2 gives the presence of enzyme to produce 6CO2 plus 6H2O. Energy 686 kilocalorie or 2870 kilojoule. Second one, anaerobic respiration. Oxidation of complex organic molecule in absence of oxygen to produce ethanol, lactic acid, carbon dioxide and small amount of energy. In anaerobic respiration, the organic food is oxidized incompletely without oxygen being used as oxidant. Anaerobic respiration occurs in the cytoplasm and releases a small amount of energy. The common products of anaerobic respiration are carbon dioxide and ethyl alcohol or lactic acid. During anaerobic respiration, water is not produced. C6H12O6 in the presence of enzyme to produce 2CO2 plus ethyl alcohol. Energy 59 kilocalories or 247 kilojoule. C6H12O6 in the presence of enzyme to produce lactic acid plus 36 kilocalorie or 150 kilojoule. Another one, ambibolic, a metabolic pathway used in both catabolism and anabolism. In Krebs cycle is primarily a catabolic pathway, but it provides precursor for various biosynthetic pathway, thereby an anabolic pathway too. Hence it is called ambibolic pathway. It serves as a pathway for oxidation of carbohydrate, fat and protein. When fats are respiratory substrate, they are best broken down into glycerol and fatty acid. Glycerol is converted into DHAP and acetyl coenzyme A. This acetyl coenzyme A enter into the Krebs cycle. When proteins are the respiratory substrate, they are dehydrated into amino acid by proteases. The amino acid after deamination enter into the Krebs cycle through pyruvic acid or acetyl coenzyme A, and it depends upon the structure. So respiratory intermediates form the link between synthesis as well as breakdown. The citric acid cycle is the final common pathway for oxidation of fuel molecules like amino acid, fatty acid and carbohydrate. Therefore, respiratory pathway is an ambibolic pathway. Another one, ATP synthetase, an enzyme complex that catalyzes the formation of ATP from ADP and inorganic phosphate. The higher proton concentration outside the inner membrane, proton return to the matrix down the proton gradient. Just as a flow of water from a higher to a water level can be utilized to turn a water wheel or a hydroelectric turbine, the energy released by the flow of proton down the gradient is utilized in synthesizing of ATP. The enzyme ATP synthetase synthesizing ATP from ADP and inorganic phosphate using the energy from the proton gradient, that is called chemi-osmotic hypothesis of Peter Mitchell in the year 1961. Another one, carbon dioxide compensation point. Atmospheric concentration of carbon dioxide at which photosynthesis just compensate for respiration. Under normal condition, a green plant accumulate organic substance beside utilizing them in respiration. The green cell must therefore build up more organic food during the few hours of light than in broken down in respiration by the entire plant in all the 24 hours. Photosynthesis is therefore a more rapid process than respiration, approximate the ratio 10:1. Photosynthesis being a more rapid process utilizes all the carbon dioxide liberated in respiration, that is, and some more from the air, and liberates more oxygen than can be utilized in respiration. Under these conditions, the gaseous exchange characteristics of respiration cannot be observed even through respiration is proceeding normally. However, at a certain low concentration of carbon dioxide and non-limiting intensity of light, the rate of photosynthesis of given a plant will be equal to the total amount of respiration. So that true respiration plus photorespiration. The atmospheric concentration of carbon dioxide at which photosynthesis just compensate for respiration is called carbon dioxide compensation point. Next one, cytogram. Hemi protein serving as electron carriers in respiration, photosynthesis and other oxidation reduction reaction. In electron transport chain, the reduced ubiquinone, that is called ubiquinol, is then oxidized with the transfer of electron to cytogram C via cytogram BC1 complex, that is called complex 3, in a small protein attached to the outer surface of the inner membrane and act as a mobile carrier for transfer of electron between complex 3 and complex 4. The complex 4 refers to cytogram C oxidized, complex containing cytogram A and A3 and 2 copper centers. Another one, electron carriers. Proteins such as flavoprotein or cytograms that even reversibly gain or lose electrons. In the electron transport chain, the inner mitochondrial membrane contains some protein which that proton ion and electron transporting enzyme. The enzyme arranged in the order manner in a specific series called electron transport chain or mitochondrial respiratory chain or electron transport system. An electron transport system is a series of enzyme and cytogram in the inner mitochondrial membrane that take part in the passage of electron from a substance to its ultimate receptor. The electron carriers includes flavin, iron sulfur complexes, quinone and cytogram. Most of them are prosthetic group of proteins. Next one, fermentation. The oxidation of carbohydrates into ethanol and carbon dioxide by certain microorganisms under anaerobic condition. While studying the alcoholic fermentation of glucose by yeast, Pasteur discovered that it can respire in absence of molecular oxygen. This process was termed as anaerobic respiration. Anaerobic respiration is an enzyme-controlled partial breakdown of organic compound, that is called food, without using oxygen and releasing only a fraction of energy. Therefore, end products are never completely inorganic. It occurs in root of some water-lodged plants, certain parasitic worms, animal muscle, and some microorganisms, example mold and bacteria. The term anaerobic respiration is often used in connection with higher organisms. In microorganisms, anaerobic respiration is often called fermentation. Fermentation is named after the product like alcoholic fermentation, lactic acid fermentation. Buchner in 1897 found that the fermentation could be caused by mixing sugar solution with the yeast extract instead of leaving yeast cell. The enzyme complex present in the extract was named as cymase. Because of the later, fermentation is also called zymosis. Another one, glycolysis. The catabolic pathway by which a molecule of glucose is broken down into two molecules of pyruvate. Glycolysis, it is also called EMB pathway because it was converted or discovered by three German scientists, Kutzau Emden, Otto Meyerhoff, and J. Pernes. Glycolysis is a common to both aerobic and anaerobic mode of respiration and therefore called common pathway. The glycolysis is the first stage of in the breakdown of glucose and in common to all organisms. It occurs in cytosol and result in the breakdown of glucose or similar kexose sugar into two molecules of 3-carbon compound pyruvic acid, releasing some energy, that is as ATP, and reducing power as NADH2. In plants, that is glucose is derived from sucrose, that is which is the end product of photosynthetic carbon reaction, or from storage carbohydrate. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides are readily enter the glycolytic pathway. The steps of glycolysis: phosphorylation, isomerization, phosphorylation, splitting, dehydrogenation and phosphorylation, dephosphorylation, that is called ATP formation, isomerization, dehydration, dephosphorylation, that is called ATP formation. Another one, accelerative phosphorylation. Synthesis of ATP with the help of energy released by the oxidation of reduced coenzyme during respiration. The oxidative phosphorylation is the synthesis of energy-rich ATP molecule with the help of energy liberated by oxidation or reduced coenzyme like NADH2 and FADH2 produced during respiration. The enzyme required for the synthesis is called ATP synthetase. It is present in F1 or cat phase of F0, F1 or elementary particle. The particles are located in the inner mitochondrial membrane. The enzyme ATP synthetase becomes active in ATP formation only when there is proton gradient having high concentration of proton on the F0 side outside as compared to F1 side, that is called inside. Next one, respiratory quotient. The ratio of volume of carbon dioxide released to the volume of oxygen consumed during respiration. So the arc is equal to volume of CO2 evolved by volume of oxygen absorbed. Respiratory quotient is determined with the help of apparatus is called Respirometer. The value of respiratory quotient varies with a different substrate utilized in respiration. Its value can be 1, 0, more than 1 or less than 1. Respiratory quotient equal to unity. Example, carbohydrate. Respiratory quotient less than unity. Example, fatty acid. Respiratory quotient 0. Mostly seen on succulent plant. Respiratory quotient more than unity. That is called organic acid. Another one, substrate level phosphorylation. Phosphorylation of ATP or some other mononucleotide diphosphate directly forms a metabolite. In glycolysis payoff phase, direct transfer of phosphate moiety from substrate molecule to ADP and is converted into ATP is called substrate phosphorylation or direct phosphorylation or transphosphorylation. Another one, additional information. Deutrochet coined the term respiration. Next one, Gruig Shang in the year 1897 coined the term fermentation. Next one, Kosti Chew in the year 1902 coined the term anaerobic respiration. Next one, Como-fermentive fermentation, when only one main product is formed. Fermentation literally means the brisk of effervescence. Next one, Ketro-fermentive fermentation, when two or more than two type of products are formed. Another one, lactic acid and heart. Heart muscles have enzyme to convert lactic acid into pyruvate. During heavy exercise, the lactic acid is formed in large quantity by skeletal muscles. The same is poured into blood. A pair of it used by heart muscles in order to obtain extra energy for rapid activity. Next one, electron transport chain poisons. One cyanide prevents the transfer of electron from cytogram A3 to oxygen. Second one, antimycin prevent the electron transport between cytogram B and cytogram C1. Third one, 2:4 dinitrophenol allow electron transport but prevents ATP synthesis. Next one, proton and ATP synthesis. According to modern physiologists, the number of protons transferred to cytosolic side of inner mitochondrial membrane is 10 per electron pair from NADH, that is 4 plus 2 plus 4, and 6 per electron pair from FADH to 2 plus 4. 3 protons are used for the synthesis of 1 ATP. However, 1 proton is required for transport of each NADH is 2.5 and for each FADH2 as 1.5. The total number of ATP molecule produced per glucose molecule is then 30 or 32 instead of 36 or 38. Another one, smallest motor. It is a biological motor consisting of parts of F0 subunit unit or elementary particle and a stalk that passes into F1 subunit. Next one, Pasteur effect. It is the reduction in consumption of respiratory substrate when the mode of respiration is changed from anaerobic to aerobic. First discovered by Pasteur, that is called Pasteur effect. Next one, Respirometer. The instrument used for measuring respiratory quotient as well as the rate of respiration.